Okay, let's go. So let's finish up the MHC Class 2 story here about how we load our peptides on there. All right, so the last couple of pieces you're going to need are, pardon me, uh, HLA-DM, HLA-DO, and then there's going to be an HLA-DR as well. And DM is going to be the loader. It's going to load things into MHC Class 2. And it actually resembles an MHC protein. And the DM is going to be the loader, and then it's going to bind to either DO or DR. DO is going to be like, oh no, don't load that thing, and it's going to block it. DR is going to be the one that says, right away, let's load things, basically. All right, so here we go. So there you got your early, so this is our little early MHC Class 2 compartment. We got our full on invariant chain here, and so we're going to cut that down to lip 22 and then lip 10, and then eventually we're going to cut it down to the clip. There we go, as we are acidifying. So the red here means we've acidified. Meanwhile, we've taken in some peptides and we've chopped them up with our cathepsins and our acidity, and we're merging those down here. And then what DM is going to do is it's going to bind to our MHC Class 2, open it up, pop out the clip, yeah, right, just like in a first-person shooter, and it's going to load in a little peptide instead. So yeah, pop out the clip and put in a peptide instead. So that's the role of DM. And so DM would normally be editing the peptide, so it can, I believe, trim the peptide a little bit. And let's see, DO is going to block DM from editing the peptide. And then DR is going to be like the antidote to DO. So DO says, oh no, don't do that. And then, yeah. So here is the little peptide. And it's going to be pinched right between the grooves of DR. It has an alpha and beta chain because, you know, a lot of proteins that we've been talking about have alpha and beta chains. And so this is going to be the loader. We are going to load our peptide on here, and then the HLA DM and DR are going to together edit and load our peptide into the Class 2 molecule. All right. Last little bit for understanding Class 2 loading. It's a complicated process, but yeah, I think it all makes sense. So we're going to have E3 ubiquitin ligases. You remember ubiquitin ligases, right? They're going to make those chains of ubiquitin. Yeah. So fine. And E3s are the ones that actually make the chain. E1 and E2 kind of set it up so that the E3 can do its job. Important protein is called March1, and this is an E3 ligase, so it's a thing that's going to make ubiquitin chains. And so what do we care about this? Well, this is going to control how long the MHC Class 2 molecules can hang around. And so it's going to, when we make March1, it's going to cause MHC Class 2 molecules to get shredded and not show anything on the outside. So this is basically a way to prevent us from showing self-proteins. Now, when there's actual danger around, when we get something like a TLR signal, then we're going to turn off March1, and so now there's going to be nothing stopping our Class 2 molecules from getting loaded and going out and presenting their peptides. Okay, next one is CD86, which is going to bind to a thing called CD28 on T cells. And it's also called B7-2. So let's go see these guys in play. All right, so we got our TLRs up here. If they pick up a sign of danger, then we're going to basically not do the things that we're showing here. So when the TLRs are not activated, so when they are both separate, they're not cross-linked, they're not signaling, And you remember all the different TLR signaling pathways, right? Yeah. There was MyD88, there were all those other things, and then we would eventually signal. Well, in addition to transcription factors making genes, this TLR signaling is actually also going to repress production of the March1 gene. So it's nice. The activation of the TLR can, block the March1, which is in turn blocking the MHC class 2 from showing its peptide. Yeah, boy, complicated, but we got there. All right, so what March1 does is, remember, it's a ubiquitin ligase. And so MHC class 2 is sitting in here, it's getting loaded up with either little clips or with little peptides. And March1 is just going to be attaching little kick me signs to the back of it, basically. Only it's a degrade me sign because it's a particular polyubiquitin chain. And you remember which ubiquitin linkage leads to degradation mostly? It's the lysine 48, K48, right? It's I think it's written right down there. There you go. And so, yeah, that's a big chain. And then we're going to take this thing and we're going to shred it. So when there's not danger, we don't want to be presenting peptides on MHC class 2. But when there is danger, then we definitely want to. And so no danger, this is what happens. March1 is basically, looks like a little hairpin and just, stops the party before it can even start. I think of March 1st as like when the rent is due. And I think of this whole thing as like a going out metaphor. You got your lip and you got your clip just like that. And then March1 is the biggest downer that's going to prevent you from going out. But if you can get a TLR signaling, then March1's going to go away. And your DM is going to load up a special peptide right here into the MHC class 2 groove. And so this is being activated by LPS. So can you figure out which TLR that is? Yeah, you remember back to our TLRs? It's going to be TLR4, right? And remember, TLR4 was the one that was able to signal the most different ways, different pathways. And so I don't remember which one of these pathways, but one of them is actually going to shut off the March1 gene. And so with no March1, and it breaks down itself pretty quickly, then the MHC can go to the outside and we can get proper loading. So that's the entire MHC story. As split up over several different slides. So we started all the way back here. We had our processing, so that's a good slide to look at. And then we were here. This is our how we get the MHC class 2 molecule ready. And then here's where we store those MHC class 2 molecules. And then here is where we're going to have that HLA-DM that's going to help us load things up when we merge our little class 2 MHC compartment with our little early endosome, or actually late endosome because it's acidified. And then over here, we've got the rest of the story. So if we cross-link our TLRs, then we will inhibit the March1 gene from being expressed. And if we have no March1 gene, then the March1 gene can't put polyubiquitin on lysine 48 and cause our MHC class 2 molecules to get stuck and basically recycled instead of showing little peptides on the outside. So there you go. It's quite a story, and thankfully, that is the end of the MHC class 2. So far in this chapter, we've covered class 1 loading and class 2 loading. Those are both totally exam questions. I think the rest of the chapter, we're going into some of the context around these. So it's not directly answering an exam question, but it's important for figuring out how this stuff works. And I think you'll be able to use some aspects of this in your answer to make your answer look even better. That's the goal. So thank you very much. That's the end of this section.